check. So I hope that you see now the PowerPoint. I see some nodding.
That looks good. Yeah, perfect.
And I will move you here a little bit so I have no interference. Okay, so thanks again for the invitation and the kind introduction. Very happy to and honored to be virtually here. Before I get started, let me acknowledge those who actually did the work. Martin Göttlich, who is still working in my group, has been involved in most of the studies. Yasmin Tolley did her master thesis with me and then contributed mostly to one of the studies I will present in the end of the talk. And Marcia Boades and Friederike Bayer both did her PhD with me a while ago. They are both now faculty in Universitat de Barcelona and at the Queen Mary University in London. So what is affective behavior about? Affective behavior refers to actions or action tendencies that are driven by emotional states or feelings. And these behaviors are therefore closely tied to an individual's affective experience, and they are often responses to internal emotional states or external stimuli. And in my group, we mostly focus on affective behavior that is shown in response to social threat stimuli, such as angry facial expressions or interpersonal provocation. And affective behavior to social threat can be studied in terms of rather basic action tendencies, like in terms of approach avoidance behavior. But it can also be studied, of course, in more complex social interactions in which threat or provocation may elicit reactive aggressive behavior. And affective behavior plays a central role in mental health and overall well-being. And altered affective behavior, in particular, also altered affective behavior to social threat, is characteristic of different psychiatric disorders, including not only affective disorders, but also personality disorders, such as antisocial personality disorder or borderline personality disorder. And many of these psychiatric conditions show prominent sex differences in terms of prevalence, but also symptoms, and which raises the question, what causes these differences between the sexes? And so you might wonder why the title of the talk is not sex differences in affective behavior then. So to explain this, let me maybe take a step back and briefly introduce another CRC which focuses on the topic of sex diversity, the CRC 1665, which has recently been established at the University of Lübeck and other collaborating universities. And the CRC sex diversity aims to investigate the determinants, meanings, and implications of sex diversity in sociocultural, medical, and biological landscapes. And the research agenda of the CRC follows the idea of sex contextualism, which has been suggested by Sarah Richardson. And the basic hypothesis here is that sex is diverse and it manifests itself through context-dependent differences that emerge on multiple different levels. And these are shown here. So the sex manifests itself on the level, starting from the level of genes, but across cells and organs up to the whole human, to the level of humans, and also affecting, of course, societal aspects like law or clinical cultures and in terms of medicine. And on that means that when we want to understand what sex means and what the determinants and implications of sex and sex diversity are, we need to consider the level on which sex and sex-related factors play a role for a given phenomenon. So whether we talk about sex differences in terms of genes or cells or organs, etc. And crucially, on some levels, sex might be binary or mostly binary. On others, when it comes, for example, to hormonal variation or functions, organ functions, or human behavior, the sex will be rather a continuum or a spectrum. And of course, it also becomes more complicated to speak about sex differences or what sex actually means, as these different levels can, of course, also interact and cannot be considered fully independent. So when we speak about sex differences in affective behavior, of course, there can be affective behavior can, of course, be affected by sex on many different levels, societal factors could play a role here for sex differences in affective behavior, different societal influences on gender roles or gendered behavior. But at least for experimental psychology and from a neuroscience perspective, the level that seems best amenable to study is the level of hormones that play a role already during brain development and then as modulators of brain function. And as you might know, although hormones such as testosterone or estradiol, which are shown here, are related to sex, I prefer to avoid the term sex hormones. I don't speak of sex differences in affective behavior or the role of sex hormones in affective behavior, because these hormones don't have a sex to begin with. They also are mostly, but are only produced by the gonads. And although the hormonal levels differ between sexes, they also depend on many factors which are independent of sex, such as age or exercise, or metabolic changes, such as in obesity, for example. So to avoid any implicit associations coming with the term sex hormones, I prefer to use the term steroid hormones and then ask what is the role of steroid hormones in these behaviors. And can these explain maybe why there are in the end sex differences in terms of prevalence of affective disorders? So what are then the neural networks which are involved in affective behavior to social threat and that could be modulated by steroid hormones? To start with action tendencies and how to study action tendencies in response to social affective signals, this behavior has mostly been studied using or has been studied a lot using the approach avoidance task. In this task, participants are typically presented with some kind of social affective signals like emotional faces, and they are told to move a joystick to either push away this stimulus or pull towards them as a measure of approach or avoidance behavior. And the typical finding here is that people are faster to push away, so to avoid negative stimuli like angry faces in this context, and to pull towards them positive stimuli, or here happy faces. And this is also shown here in the bottom. This is the typical approach avoidance response bias, that one is faster in avoiding angry faces here and faster in approaching happy faces. And the group of Karin Röhloffs has in many studies delineated the neural circuits that are involved in this kind of automatic affective behavior and its regulation. And this is summarized here in a review paper by Calderay and others. Central for this in this model here are interactions between the amygdala, which is relevant for early processing of these affective signals, and then interactions with the anterior PFC nodes together with other areas of the frontal parietal network. So the idea here is that the anterior prefrontal cortex is especially important in regulating and overcoming automatic affective behavior in this kind of task, and ultimately then regulating approach and avoidance behavior in this setting. Also in our own studies using this approach avoidance behavior task, we could show that the OFC also seems to play an important role in the regulation of affective behavior. Here, patients with OFC lesions due to a head trauma or tumor showed altered affective behavior, and in particular, they showed increased approach tendencies to angry facial expressions. In a series of studies, the group of Karin Rüllos has already demonstrated that especially testosterone, also in interaction with cortisol, plays an important modulating role here for this circuit. So they have shown, for example, that testosterone modulates the connectivity between anterior PFC and amygdala, which resulted in a negative correlation between testosterone and prefrontal inhibitory control. They could also show that testosterone administration in women leads to reduced threat avoidance on a behavioral level. So they demonstrated already that testosterone seems to have an important role and influence on approach avoidance behavior to social threat signals in particular. What about more complex behaviors and reactive aggression in response to interpersonal provocation? The network involved here in reactive aggression was also more complex, as can be assumed. This is summarized here in a recent review by Katja Bertsch and others. Again, here at the center, there is again a basic threat network comprising the amygdala, hypothalamus, and periaqueductal gray, which is highlighted here in red, and which is important for triggering aggressive behavior. But in addition to this threat network, there are other networks which play a role, networks which are involved in evaluating reward or frustration, which also often triggers aggressive behavior. But of course, these networks are also then regulated or presumably controlled by prefrontal and frontoparietal networks, including not only dorsolateral prefrontal cortex, but also ventral, especially ventromedial or orbitofrontal areas. How can we study these networks within human neuroscience research? So in our lab, we adapted an aggression paradigm from social psychology that was suggested in the 60s already by Taylor. And this paradigm is set up as a simple competitive reaction time task in which participants are playing in multiple rounds reaction time task with a putative opponent. They, in fact, play against the computer. And in this game, the winner is entitled to punish the loser using an aversive noise. And interpersonal provocation here is then manipulated through increasing punishment selections, that is, louder noise levels by the opponent, and this is expected to elicit aggressive retaliation by the participant. Here we extended this paradigm to study especially the neural and behavioral response to social threat signals, also on a very basic level in terms of angry facial expressions. And so what we did is that we told participants they would see their opponent at the beginning of each trial via webcam. In fact, they were presented pre-recorded videos. And in these videos, the opponent would either make a neutral facial expression or make an angry facial expression. And this adaptation allowed us to then investigate whether these social threat signals add to the provocation in the reaction time game, and whether inter-individual differences in the neural response to these threat signals relate to aggressive behavior. And as can be observed here, on a behavioral level, as expected, the aggressive behavior shown by the participant increases over time with increasing levels of provocation by the opponent. And this is even more so the case when the opponent makes an angry facial expression. So this additional social threat leads to even higher levels of aggression in the participants. So what are the neural mechanisms here, or the neural networks that play a role here? So in terms of neural activity, the comparison of angry and neutral facial expressions elicited activity in parts of the network that we have just seen in the review paper by Katja Bertsch, like medial, dorsomedial and ventromedial, prefrontal cortex, also insular, and also areas of the temporal cortex. Importantly, the amygdala reactivity to angry facial expressions as compared to neutral facial expressions was across participants related to the level of aggressive behavior, such that those who showed higher amygdala reactivity to angry faces, they also showed more aggressive behavior during the task. So similar to what we've seen, or what the idea is behind the approach avoidance behavior, the approach avoidance task, we also here see a positive relationship between amygdala response to social threat signals and aggressive behavior. But the amygdala is not the only region that is relevant here. Amygdala and OFC showed reduced connectivity when being confronted with social threat signals. And in addition to the amygdala, we also saw a positive relationship between the superior temporal gyrus and aggressive behavior. And mediation analysis showed that the relation between the amygdala response and aggressive behavior was fully mediated by the superior temporal gyrus activity. So that suggests that the amygdala response to social threat then modulates the STG response and in the end favors aggressive retaliation. So then the question is, how is this modulated by endogenous testosterone levels? Does that modulate the neural and behavioral response to social threat signals also within such an aggressive encounter? Yes, it does, but surprisingly, we actually found in this sample of 39 women, we found a negative relationship between endogenous testosterone level and aggressive behavior. So in contrast to what is usually, quite often, at least in men, reported, that there's a positive relationship between testosterone and aggression. Here we found a negative relationship, which still is consistent with other experiments which demonstrate that, at least in women, the role of testosterone is more complex and not always favoring aggressive behavior, but might also favor pro-social behavior, dependent on the context, if it's helping one's own status, for example. What about the neural response? We also found a negative relationship between the testosterone level and the amygdala reactivity. So again, here the amygdala reactivity was higher for those who had lower testosterone values. And one could assume here that this amygdala effect is mediating the relationship between testosterone and aggressive behavior, but this was not the case, at least as far as the mediation analysis suggested. So this suggests that there are other factors which are here still in play to explain how testosterone directly influenced aggressive behavior. So while these studies related testosterone level to the neural and behavioral response to social threat in a more state-like manner, like acutely, testosterone might also play a role in a more trait-like manner. And especially for a long time, there has been the hypothesis that testosterone, as I also mentioned briefly in the beginning, that testosterone androgens have an important role in brain development already, so-called organizational effects, and not only activational effects, that is, not only state-like effects on brain function. Of course, these organizational effects, like effects during brain development, cannot easily be studied here. But one first step would be to see, well, are there many maybe more stable factors like functional connectivity or structural connectivity patterns that are associated to testosterone and that are independent of the social interaction and the direct provocation. So is endogenous testosterone associated with a functional and structural connectivity pattern that might predispose to reactive aggression? And so to study this, we used the same data set, but here we measured functional connectivity in a resting state measurement before the actual aggression paradigm. And we found that testosterone was negatively related to connectivity between the amygdala and the basolateral nucleus group of the amygdala and the superior temporal gyrus. And the same connection between amygdala and superior temporal gyrus was then positively related to aggressive behavior. So again, as we've seen also in the functional in the fMRI results during the task, we see the negative relationship between testosterone in the end and the neural mechanisms that are positively related to aggression. So this node in the superior temporal gyrus was not really expected. It's not really part of the typical network that we would assume for regulation of aggressive behavior. But it's at least consistent with some studies which show or suggest that this region is also involved in rather multimodal threat detection. Of course, we cannot directly interpret this here as this is only resting state data. But at least these data suggest that functional connectivity between the amygdala and superior temporal gyrus might underlie the hostile readiness or related to the hostile readiness in an aggressive encounter, and that this is blunted in women with high testosterone levels. What about structural connectivity and not just functional connectivity in these resting state measurements? So we also examined structural connectivity and especially structural covariance. Structural covariance, here the covariance between gray matter volume in different brain regions is supposed to reflect joint development or sustained co-activation, and can therefore be a proxy for structural connectivity. And so we analyzed in separate samples in both male and female participants. We analyzed the relationship between structural covariance of the main amygdala nuclei groups with cortical regions, and whether this structural covariance is modulated by testosterone levels. And interestingly, here we actually found those regions which we would also expect based on models like the one presented by Katja Bertsch. So here we find a structural covariance between the centromedial aspect of the centromedial nucleus groups of the amygdala and the dorsomedial part of the prefrontal cortex. And this was found to be related or modulated by testosterone levels in male participants. In female participants, on the other hand, we found a different pattern here, the connectivity or the covariance between the centromedial nucleus group of the amygdala and the ventromedial part of the prefrontal cortex was modulated by testosterone levels. So these are relatively small groups for this kind of analysis and awaits further replication. Also, as this was from different studies, we could not directly compare it. But of course, it would be interesting to see whether this is actually a robust finding, which would suggest that these effects also interact with other sex or gender-related factors, leading to different patterns here in men and women. But again, these should be replicated in larger samples to see whether they are robust. So coming back to the beginning, we can already conclude that there's evidence for modulating a role of testosterone on approach avoidance behavior and aggressive behavior and reactive aggressive behavior. And that this modulation is also found on a neural level in terms of amygdala activity and structural and functional connectivity in amygdala-dependent networks. But what about other steroid hormones than testosterone? What about estradiol or progesterone, for example? And as you all know, levels of estradiol and progesterone are changing throughout the menstrual cycle, and they are also strongly affected by usage of hormonal contraceptives. So the endogenous levels of estradiol and progesterone are strongly reduced in women taking oral contraceptives, and there is no modulation. The level is flattened and stays the same across the cycle. What the effects are of this are not well understood. We know that the prevalence of adverse effects, for example, on mood and affect, range between 4 and 10%. There seems to be higher prevalence of adverse effects in girls or women who have already some risk factors, which can be expected. But generally, we know little about what exact cognitive and affective processes and also what neural processes are effective by taking oral contraceptives and by thereby changing the endogenous levels of these steroid hormones, and also for affecting the changing the fluctuations which usually happen during the menstrual cycle. So how can oral contraceptives affect brain function? There are different ways how they can affect this. So first of all, the synthetic estrogens and progestins could bind to endocrine receptors and thereby affect brain functioning. The reduction of the endogenous levels of estrogens and progesterone could have an effect, of course, on brain function. And there could be also indirect effects in terms of effects on neurotransmitter systems, like dopamine or other endocrine receptors, like effects on androgen level or interactions with glucocorticoid or mineralocorticoid receptors. In a recent review, Montoya and Bose have summarized the brain systems that are possibly underlying the effects of OC, the usage of oral contraceptives, on social, emotional behaviors. And so these networks, which are shown here, could also potentially contribute to adverse side effects in terms of changes in mood and affective behavior. But what exactly is altered in terms of cognitive or affective processes and behavior is still not known, or relatively little is known. And so as these are networks which are also implied in approach avoidance behavior and aggressive behavior, we started by looking at how approach avoidance behavior is altered in oral contraceptive users. And so in this study, we had 65 women who were taking oral contraceptives, combined oral contraceptives, and compared them with 65 women who were not taking any hormonal contraception. And they participated in this study, which had several parts. They did the approach avoidance task, which I explained already twice. And in between, they had a stress induction. So the socio-evaluative cold pressor task. So they were asked to put their hand for 3 minutes into ice cold water, which elicits stress. And the stress is even stronger by having the social evaluative component, which means that someone is looking at them, evaluating them, directing a camera at them and recording them while they are standing there and putting the hand in the ice cold water. And this procedure has been shown to reliably induce stress and also cortisol increase. And as the steroid hormones, which are affected by oral contraceptives, could also interact with cortisol responses and thereby have an influence on affective behavior, we wanted to study both the effect of oral contraceptives and the potential interaction with stress responses. So the approach avoidance task was, as I presented already, and this is what we found in terms of the behavior. So what is shown here is the approach and avoidance bias for the two different groups and the two different stimuli, like angry and happy faces, and before and after stress. And what we found is that OC users showed a reduced or actually fully abolished social threat avoidance bias, which can be seen especially on the right side, where it's average across the conditions before and after stress. So this was not modulated by stress itself, but generally there was no avoidance bias anymore in the OC users, as we found in the non-OC users. And at the same time, we found a stress-induced increase of the approach bias. So OC users generally showed an approach bias to positive stimuli, to happy faces, but this was even increased after stress, and this was in contrast to the non-OC users who did not show any changes due to the stress induction. So this demonstrate that oral contraceptives change or affect already affective behavior on a very basic level of automatic affective responses. We have this was a purely behavioral study, so it awaits replication and further neuroimaging studies to understand which are the neural pathways through which these effects here happen. In the end, I would like to give an outlook what future research should look at, or what we also want to look at in future research. As I mentioned already, there's the long-standing hypothesis that androgens have an important influence on brain development already. Which is, as you can imagine, difficult to study in humans, where the levels of testosterone or androgens, generally of steroid hormones, prenatally is usually fully confounded with the hormonal levels postnatally, and then also with societal influences which then shape gender behavior. So we cannot disentangle at all anymore which are effects of socialization or effects of genetically determined hormonal levels during brain development. But people with variants in sex development, with so-called differences in sex development, DSD, might present an interesting case here to study actually the role of genetically determined prenatally altered hormonal status, which is then relatively normalized after birth, so that we can see what the effects are, especially of these prenatally altered hormonal levels, for example, in people with congenital adrenal hyperplasia, and can see how this affects brain function and also behavior later on in life. It's also important to take a broader picture and consider a broader picture and not only measure specific hormones, as I showed here in these studies, which only focus on testosterone or estradiol. So to consider steroid hormone profiles would definitely be interesting in the future. And of course, also behavioral profiles to have a more comprehensive picture of how specific also some effects are in terms of behavior. And with this, the take-home messages kind of summarized are that endogenous testosterone is associated with amygdala activity to threat signals. And we could also show that functional and structural connectivity in amygdala-dependent networks is modulated by testosterone levels. We also see that in users of oral contraceptives, approach and avoidance behavior to social threat signals is altered, which might contribute to also adverse effects of taking OC, taking oral contraceptives. I also think that oral contraceptives present a quite unique lens or an interesting method to study hormonal modulation of social and affective behaviors. In the study that I presented, this is a cross-sectional design, so of course this is suboptimal, but in principle it is also possible, and of course advantages in the future to also study participants before they start taking oral contraceptives, and then see how taking the oral hormonal contraceptives changes their behavior, affects their behavior when taking them for a few months. And generally, of course, this is more in the very long run. I think this kind of research, of course, can have implications for mental health, as it can give the potential for personalized approaches by targeting hormonal pathways in people with affective disorders, for example. And with this, thanks to the members of the Institute of Medical Psychology, thanks to the DFG, and thanks for your attention.